Hi, this is Eli Ogletree of the SnoKing Beekeepers Association, and this is our June 16th, 2021 monthly meeting every third Wednesday. And thank you for joining us or for watching this this YouTube video later. And we'd like to put up our informational slide at the beginning so that people can find us, have an idea of how to contact us. We are going to go through some real quick announcements, just that we are a club. There are a number of people who are joining us tonight who aren't particularly associated with our club. Our club is loosely affiliated with each other. Mainly we meet on the discussion page on Facebook. We don't have a due system. We do ask when you take a class that you pay costs, so I put that up. We have a Bee Buddy system instead of a mentor system. We're finding it's easier to get two people together who know something about beekeeping, are going through the experience themselves, than to try to put one person in charge as the experienced mentor. It's working better. And call me. I'm happy to answer questions anytime. And same for Ron and a lot of other people. And on the discussion page, you can make connections there too. Our high side chats, they're not really chats, they're hands-on. We're not taking more than about 5 people, and you can actually hold the hive tool, hold a frame of bees. This is actually really good for people that have never had bees before, or this is their first season. And if you're more experienced and you want to come, it's wonderful. We love having the discussion and the comments. And yeah, I would scrape all that propolis off. No, I would let it sit. I think you've got this, that. It's really kind of neat. So we have found that it really works well. There will be Hiveside chats this weekend. My place on Saturday at 1 p.m. close to Maltby is the best I can give you my location. I'm in the middle of nowhere. Ron is Granite Falls, and it's Father's Day, I know, on Saturday, but if that is your Father's Day dream to go out and work in a beehive, get a hold of Ron. He said both phone and email, and I didn't get them put up here. I can put them in the chat though, and they will be on the website. And we don't, anything is donation, and people have been donating. Extractor rental, we have this one little extractor, in case you've gotten to that point, and that is donation too. Little 2-frame hand crank. And this is the YouTube channel that we will be putting this recorded meeting on. And please subscribe while you're there. We are on the edge of 100 people. When we get 100 people, we get to name this channel. Otherwise, there's this terrible little string of letters and numbers up here. And that's our name right now. We hit the magic 100. It should happen this week. You could be number 100 if you subscribe while you're going to view this meeting or past meetings. Okay. And this is my last slide of announcements. A lot of times I tell people it's under the files on the closed discussion group page on Facebook. It's closed to people who are not in Western Washington, and we allow a few other people in because they're advanced journeymen and such, and they help out our club with comments and help out the discussion. But pretty much we want to be Western Washington. And we love that they're doing beekeeping in Nigeria or Venezuela or wherever, but we really want to focus on our needs and our interests right here in Western Washington, which is actually very different from even Eastern Washington. So there's a files there. And for example, the PDF, the trapping PDF, which you can find online very easily, but just in case you're already in the discussion group, you can go to files and click on the trapping instructions there. Because, and Cassie's going to talk about that as soon as I finish this page. Bee Buddies and Hiveside Chat. Oh, classes and certificates. Congratulations to 6 more beginner and 7 apprentice grads. And we're still doing online classes, and the tests are online now too. The next beginner class starts the beginning of July, the 1st or 2nd week. I haven't quite set that up yet. We are working on a new website, which will get us an additional forum in addition to Facebook, which has worked okay. So Cassie's going to talk, and I think she just wants to do a quick 10 minutes on this kits that are being made available. You don't have to belong to our club. We'll help distribute anything in this area. And we don't have a definite club membership, so we're quite happy to do that. Actually, I've already had 7 kits requested. So Cassie, we might have to ask for more because we only asked for 15 before the news broke on the major media. And then Rex is going to do the talk we're really waiting for, the induced brood breaks for Varroa control, because we are all hoping for better control of Varroa, fewer chemicals. We are looking 34 years with this pest. So Cassie, do you want to go ahead?
Awesome. Thank you for the introduction.
Yeah.
I work for the Washington State Department of Agriculture. If we haven't already met on one of these meetings, I work obviously on the Asian Giant Hornet Response Project. And if you saw any of the news today, an Asian Giant Hornet was detected near Marysville. So I'm reaching out to your Bee Club Association and asking if you're interested in being a cooperator with the Washington State Department of Agriculture and trap for Asian Giant Hornets. So basically you still follow the citizen scientist trapping program, but I am able to provide the trapping supplies for you. So basically I just will get a number from you guys on how many traps you want and how many people, how many traps you want and how many people will be running the traps. I'll get all your supplies together and I'll drop them off in one central location so you can come and pick them up. And that's basically it. Basically what the trapping program is, it's a 22-week long program starting in July and running through November, or as long as you can trap. Like if you go away on vacation, just take your trap down. When you come back, put it back up. What these traps are is essentially just a 2-liter clear plastic bottle. And then it has a bait solution in it. And the bait solution is rice, cooking wine, and orange juice. And we picked that based off of what the trappers over in its native range in Asia use. And it's pretty attractant to the hornets. It doesn't attract your honeybees or our native bombists and other bumblebees, that we have. So it's really good for that. Essentially what you'll do is you'll get your trapper, how many ever traps you decide, and you'll find a place that you have access to, trap on your own property. You'll hang it up in a tree, and then each week you'll service your trap. What servicing is you go to your trap, take it down, you'll dump the contents into a strainer, look through your insects. If you don't have an Asian Giant Hornet, discard it. If you think you might, just snap a picture and send it in an email, and we'll follow up with you after that. And then you just rebait your traps, so pour the rice, cooking wine, and orange juice back in it and hang it up. And it's essentially just that. So with that being said, if you guys have any questions, I'll take them. I guess the other thing I should mention, if you are hanging them near your apiary, they just need to be about 50 feet away.
How many traps per acre?
There's really no definite answer. I mean, any traps are going to be extremely helpful. So I would just do what you think you can handle. Like if you think you can handle 1, that's great. If you think you can handle 2, that's great too. So whatever you guys are willing to commit to, we really can't be more thankful for it.
So I live in Snohomish, but we have a farm up in Washington County too. Should I put traps up there?
If you would like to, I can definitely get you the supplies for both.
Okay.
Basically right now, obviously we're targeting Whatcom County and then with this new find in Snohomish, but also anybody that lives in Skagit, Clam, Jefferson, any of the island counties. And now, we're kind of looking into King County too, are definitely more than welcome to trap. Any resident of Washington really is. We're just kind of, on the west side of Washington is more of a target.
Sam with it?
No, Commander.
Any other questions? And the trapping instructions are under files. Oops. Under files, as I was saying, on the group discussion page. We'll put them also on the business page, which everyone can get to. We already have a distribution point at Pat Waymeyer's in Marysville. So we have a Marysville distribution point, and we're looking for one that's farther south would be nice. I can do it. I'm Maltby, which is not bad. I'm still inside Snohomish County. Or farther.
I also think I think I had a conversation today with Corey, their Northwest District Beekeeping Association, but he owns the Snohomish Bee Company. And he's also offered their business open for people to come pick up trapping supplies as well. So we might be able to coordinate that together.
What's your contact information, Cassie?
I will throw that in the chat right now because my last name's really weird. So
It is Cichorz, right?
correct.
I got it right. Yes.
So I put in my email in there. Let me make my email and my phone number. You can send me an email, give me a phone call, shoot me a text. Sometimes I don't respond in the evenings, but I'll definitely get back to you during the day.
Anything else for Cassie? Because we appreciate her coming special just to help us set this up.
Yeah, awesome. And then again, too, like you guys are citizens on the ground. You live there. I live in span away, so I'm a ways from the action. So I really appreciate your efforts and all you guys do with just being on the lookout and reporting if you think you see one. Honestly, couldn't do it without you guys.
Thank you. Thank you for doing this. This is such an important program. We'll all sleep safe, sleep more soundly nights knowing somebody's watching.
All righty. Well, I'm going to hop off and let you guys get to the good bust of your meeting. Have a nice night, all.
Thank you. Good night.
Yep.
Okay. Is Rex ready? Do you want to share screen?
Sure.
And Nick, let's see. I sent out your bio. It's pretty extensive. I mean, your background and everything, farming, ranching, gardening, organic, biotechnical methods, and then actually working with the Washington State Department of Agriculture to get an experimental use permit for oxalic acid along the lines of what Randy Oliver was doing. I mean, this is very impressive. I'm so impressed because, I mean, this is like, to me, PhD quality type stuff. This is real research, documentation, and everything. So, okay, I should stop raving and let Rex start talking. Ready?
Thanks, Eli. Thanks for inviting me. I'm going to go ahead and share my screen. I think the best way to do this is I'll kind of run through. I've got a bunch of slides, it's a lot of stuff. And we'll do questions at the end. And go ahead and make sure that you're muted so there's no distractions for us. And I'll go ahead and share screen. Okay, is that, everybody see the giant scary mite?
Yes, we can see it.
Okay, thanks, Sarah. Okay, so a few years ago, I got interested in a lot of research that has been done in Europe, and there's quite a bit of research and a lot of beekeepers, even on a commercial basis, that are making a induced brood break part of the control of the Varroa mite. And so I've been reading everything I can get my hands on. My mentor, master beekeeper Greg Butler, who I think he's listening in here, has helped a lot. He's gives me a continuous supply of research stuff to read. Whenever a topic comes up or I ask him a question, he has been really good at helping out. So we've kind of been really doing this work together. So an induced brood break, and the whole idea is to, when it comes to that time of the year when we're raising the winter bees, to have healthy winter bees. So this topic can actually get kind of complicated, and because there's so many different ways to do it, so many variables on each method. So what for tonight, what I want you to do is just we're going to concentrate on two kind of standard techniques. And if you kind of get this in your head, kind of get a picture, then as we go along and we look at other, like basically modifications. In your head, if you can say, oh, well, that's kind of like what they do in Italy, 25-day queen caging and then oxalic acid on day 25. And then the other technique is the German, what they call the classical trapping comb with no chemicals. There's people in Germany that have been doing this for years with no chemical treatment. And this research on this came out way back in the 80s. It was developed partly to as a swarm control, but also combining Varroa control. So this picture on the left is a picture that Greg Butler provided. He's been doing this for a couple of years. So you can see in this frame, there's a little cage that's mounted. And so the queen goes into this cage for 25 days. Then they release the queen and they do oxalic acid treatment. And it could be some parts of Europe, they do dribble because they aren't registered for the sublimation, but the ideal is to do the sublimation. And commercial beekeepers are doing this in Italy. They ended up with so much mite resistance to the chemicals. They actually, some of them do this 2 times a year. And so, okay, so kind of get that picture in your head, a cage, 25-day queen caging, and then oxalic acid on day 25. So now over here is, this system was developed in Germany. And down in the box, this is a frame cage. And there's one frame in there and the queen is on that cage. This is trapping comb number 2. In my hand is trapping comb number 1. And this is day 18. Don't worry too much about the details. Just get the idea of a cage, trapping comb cage, and no treatment with miticides, no chemistry. So I'm going to go, as we go through this, I'm going to go into the details of this, so this will make sense. But for now, just in your mind, a frame cage and a series of trapping combs. So today, this is day 18 actually. So this comb that's in my hand is going to go in the freezer. I'm going to kill the mites and the infested brood. And then we're going to progress through 3 trapping combs. So for now, Italy or Germany, those 2 images. So now, as we go through this, I'm going to touch on some other, let's say, modifications, okay? So as long as you can kind of relate it to one of these modifications, then this will make a certain amount of sense and you won't get lost. So I'm going to kind of go through the presentation today, is going through some of the details on these 2 methods. And then I'm also, last year I did, as Eli mentioned, I had an experimental use permit from the state, and I did a research project, and it was basically a modification of this German classical trapping comb. And the modification was based on this research that Randy Oliver has been doing for like 5 years now. And so you can see this is a brood box. I do all my beekeeping in medium. So I have 3 medium broods and I winter in three brood boxes. So this is a lower brood box. You can see a trapping comb down here, a frame cage with a comb in it. And then this is the extended release in the shop towel. And we'll talk about some more details. And if I don't answer your questions, if you have questions as I go through, then make some notes and we'll talk about that as we go. So organic and biotechnical is important to me. It's just not an option for me to be using amatraz and the chemical treatments. And I've been gardening for 45 years, haven't had to use any chemicals. There's always a way to grow something if you're willing to, figure it out, or else just forget it and don't even try grow something else. But I have 70 blueberry plants, so I don't use any chemical treatments on them. Same with the garden and the apple trees. So oxalic acid. So as far as chemical treatments, the only thing that I use is oxalic acid. So I'm going to talk about that a little bit. And it is organic approved. This is a website for Organic Materials Review Institute. Allowed with restrictions. That's the EPA restrictions and mite control in honeybee colonies. Oxalic acid, you probably know it's in everything. It's naturally in honey. This is a photo I took of a small plate of baby spinach. That's 100 grams. That's not a dinner plate. It's one of the smaller plates. So there's probably about 1 gram of oxalic acid in that little bit of spinach. So like this winter when I did sublimations, I did 2 gram sublimations on each colony. That's not very much oxalic acid. So I know people have concerns about using, some people have concerns about using oxalic. Wouldn't it be great to not have to use anything? And then there's the issue of, having to spend all the money on the vaporizer and the mask and all that stuff. And then some people will say, well, if you got to wear a gas mask, then I'm not going to use it on my bees. And I understand that, but I think, that's really where I was at like 3 years ago. The more you learn about oxalic acid and as you use it, the more comfortable you are with it. And I really don't have any concerns about using it as a treatment. If I don't have to use it, then that's better yet. That'd be the goal. And some of my colonies, that's, I can get by with maybe a couple treatments a year. But that's kind of where I'm at on the oxalic acid. What about mite resistance? So this study was done in 2017, and they studied mite populations that were subjected to 64 consecutive treatments over 8 years. And they compared it to populations that had never been treated with oxalic acid. They were like treated with the chemicals. So 8 treatments a year for 8 years. And it turned out that the mites that had been treated with oxalic all these years, they were actually more sensitive to oxalic acid than the bees that had never been exposed to it. That kind of surprised the researchers. So I don't know what, 40 something years of use in Europe, and there's no resistance. So not a whole lot of concern about that. As far as research, this is a really good paper to look at. Journal of Apicultural Research, Summer Brood Interruption as Integrated Management Strategy for Effective Varroa Control in Europe. And this was an extensive study. It was 2 years, 10 different countries, 15 different apiaries. And one reason I recommend you check out this article is because it's written really well, and it's really easy to understand what they were doing. And this, real quickly, this is a graphic that's off of that research paper. And up here, this black line here, this is the timeline. And the start time is here at 0. There was a pre-assessment time prior to the starting. They counted mites for 90 days, daily mite drops. All of the treatments were done by day 35, and then they did a critical treatment, which means they put amitraz in there and counted out to day 75 so they could figure out how many mites they missed with the treatment and then calculate the efficacy. So at the top here, queen caging, that's the Italian method that I was talking about. Cage the queen, 25 days, chemical treatment. This bird removal, this is kind of a type of split. I won't spend time on that, but it's another, it's a variation, really a variation on a trapping comb. This is the classical trapping comb, the Germany that I showed you at the beginning, classical trapping comb, a series of 3 combs and no oxalic acid. And then down here, this is another modification. I haven't done this one, but I'm actually kind of interested because it's very simple. It's basically, and here's where the confusion comes in, because if you look at this, this is trapping comb method simplified. That's what they called it. But it's actually 25 days caging and then oxalic acid. So what they did is they caged the queen on a trapping comb for 20 days. Then they pulled the trapping comb out 20 days because any longer and the mites are going to start emerging, right? So you pull it out at day 20, or you could do day 19, destroy that trapping comb. Then the queen goes in like any kind of other cage, like, you know, the one that I showed you at the beginning, the little Italian, and you keep her for 5 more days. The whole idea is to run this out 25 days. So all the mites have emerged out of the drone brood. That's why the 25 days is important. You really don't want to mess with the 25 days. So anyways, this research paper is definitely recommended. It pretty extensive research. Also, on YouTube, you can go to the National Honey Show Lecture Series 2019. And Dr. Ralph Buchler, who is a researcher in Germany that's done a lot of work on this, he at the Honey Show, he did it was actually a series of 4. And this part 3 is the one that will kind of explain the stuff that I'm talking about. And if you go and listen to this, you'll kind of see that a lot of my information, it comes from his research, and this is kind of a summary of his research. But also check out these the other 3 presentations that he did. He's been basically spent most of his life studying Varroa resistance. He studied these populations around the world that are in the wild that are basically resistant to mites. And so check out Dr. Ralph Buchler. So this whole system, like induced brood break, we're talking about a break where there's no egg laying, usually 25 or 30 days, but in maybe June or July. And what they are doing is kind of copying what has been going on for millions of years, where the bees naturally swarm in May or June. And in the process of that swarming, there's a period where there's no queen laying in there. And so that's what this is all based on. And it's, swarming is reproduction, but it's also part of a strategy to interrupt the disease cycles and renew the nest. And our modern beekeeping method, we don't allow that. So we have this continuous brood rearing, and that favors the development of diseases and especially the mites. So one thing about this brood break, when you think about these bees in the wild that have developed resistance, the swarming and the brood break with the swarming is a major part of that. And one of the things that happens is that the physiology of the Varroa mite changes when they're on this extended dispersal stage. So the reproductive ability is reduced for a couple of brood cycles, 1 or 2 brood cycles. So not only do you deny the brood for the mites to reproduce, basically you stop the reproduction and it messes up the mites, but you also change their physiology temporarily. Just having an interruption for 25 to 30 days will reduce, will cause a mite loss of 40 to 50%. So doing nothing else, just whether it's a swarm or you're caging, no chemical or nothing as far as another treatment, you actually make a significant reduction in the mite population. You reduce the viral load. It's possible to actually increase your honey production if everything's timed right. And that's because, well, we'll get into that. We'll talk about that later, but that's a possibility. And of course, winter bee development for winter survival. And we're going to spend a little bit of time on that. So what happens in a natural swarm? This is a way of visualizing what's happening in the source colony or the parent colony. So this is just one scenario that could happen with a brood break, with swarming. So along the top, this horizontal line here, this is the timeline. So ignore all this other stuff for a minute and I'll kind of orient you to what you're looking at because it looks confusing. But so we have a timeline across the top and there's a date, and this 31, that's May 31st, and here's June 1st. So the month of June runs across here, and then there's July 1st right there. And then going down vertically, this is brood development, and it goes down 24 days because it's the drone brood that we're concerned about. So what we're looking at in this example is on the 31st, the queen laid some eggs. Let's picture that she laid some drone because that's our main concern. Yeah, of course, you're picturing the whole nest is kind of at a peak population with, eggs, open brood and sealed brood. But They made swarm cells a few days ago. And on the 3rd, June 3rd, the first swarm cell was capped and the colony swarms on that same day. You know, a lot of times they'll swarm that day or the next day, but you know, this is variable. But in this example that we're looking at, they capped a cell, the first swarm cell, and the colony swarmed. So now the mother queen is gone with the swarm. That's another story. But what we're talking about is here in the source nest or the parent colony. So looking at this timeline across the top, on the 10th, the queen emerges. Then she needs 5 or 6 days for physical maturing. And in this example, we got cold, wet weather. So she's not getting mated yet. Now, well, what else could happen? What about cast swarms? If there are cast swarms, which is fairly common, that also extends this brood breakout. So, so in this example, on somewhere around the 24th or so, the queen gets mated and the first eggs are laid on June 26th. So now over here, you see the brood developing eggs that day. By the 29th, we're seeing some open brood, and we won't see any capped brood until around the 5th of July. So they're basically, if you go from the 10th, so all this open brood was capped by the 10th. And so this brood is, it's all capped and emerging as time goes by. And in this 11-day open period, the mites come out. They have nowhere to go, so they're susceptible to grooming and just, you know, mite loss, you know, drop the mortality, and they have to make that physiological change that we were talking about. So anyways, the whole idea is this happens in swarming. This has been going on for millions and millions of years. And it's a part of the natural process. And that's, so these brood breaks are based on that. So, and down here at the bottom, it says table format adapted from scientificbeekeeping.com with permission from Randy Oliver. So if you've been on Randy's website, you've seen this. Basically, I've copied the colors and everything, this table with this timeline across the top and the development going down vertically. This is something that I've found on Randy's website, and he gave me permission to use this, and I find it really helpful for visualizing. And you know, you can do this yourself, grab some graph paper and kind of map things out if you're trying to picture what's going on in the colony. But anyways, thank you, Randy Oliver, for helping out. Now, if you go to Randy's website, he's got some good information there on brood breaks, but you want to make note, this graph or table that he did is 20 days. So this is for the worker brood, not the drone brood. And Randy, in the summertime, in his climate, he doesn't have drone in the, it's not even an issue. So you want to like make sure that you're paying attention. If you you can release the queen earlier, but if you have drone brood, you don't want to do that. But this works great for Randy in his area in the summertime because he doesn't have the drones. So he can release the queen on day 14, and then he gets a couple days of open brood and he does a dribble in there. So basic, what I say for us in Washington, Pacific Northwest, don't mess with the 25 days. You don't really gain anything. You don't have to be in a big hurry. You can cage the queen for 30 days or longer if you want it. Stick with the 25 days and you'll get more benefit. And this is just an example. If you have drone brood that's going to emerge in 24 days and you release the queen on day 14, you don't get an open brood period. You got the double overlap. So, map it out and figure out what you're doing. So, so now Italian queen caging, 25 days for a drone cycle. So last year when I did this, I did the caging in July, and it worked. The colonies, I did it on three colonies. They built up a winter brood. They wintered fine, and they came out of spring and grew really well. So, but I, studying a little more and trying to time it with the honey flow, I think it's going to be better to be doing it in this first week of June. So that's what I'm doing this year. So this is just an example of the same, timeline across the top. Release the queen on day 25, and it gives you this open window here. So for me, where I'm at, the blackberry and hopefully the snowberry is starting like right now. Like I had my first blackberry pollen coming in last Friday, I think. Yeah, I think that was the 11th. And so it's just getting started. We had cold, rainy weather, but today, with this weather forecast, it's looking really good. So anyways, middle of June to the middle of July is what I kind of, somewhere in there, hopefully there's a honey flow. So if you look what's happening here, the eggs in the open brood, they end up there by day 12, 13, 14, all of that brood is capped. So now the nurse bees, all of the food and energy that was going into raising that brood, they're done with that. They don't, they're free to move to processing the nectar and all the other activities. So this is another way of kind of visualizing that. So we're talking about queen caging. Well, so I'm also doing the trapping comb, the German trapping comb without the chemical treatment. So basically the same thing. It's just on that, I'm starting in the first week of June and releasing the queen on day 27. So basically the same thing as far as a caging period. So across the top, we have a timeline: April, May, June, July, and these are weekly days, like I think every Wednesday, the 7th, the 14th, the 21st, the 28th. So this queen caging is in this first part of June. And then the honey flow, this represents the honey flow down here. 4 weeks. So the middle of June to the middle of July, roughly, give or take. So now we can kind of visualize. So let's go here to April 14th and visualize say the queen laid up a frame of brood on the 14th. So 3 days as eggs, 6 days in the larval stage, 12 days as cap brood, 3 weeks in the brood for development. So around May 5th, basically you're just reading vertically up here. So around May 5th, that bees that result from eggs laid here are emerging on the 5th. So then they have a three-week hive period, and they begin foraging around the 26th of May. And so again, 3 weeks in the hive period. And then for the foraging period, I put 3 weeks. Several of the popular books say 3 weeks, but you'll see research that say it can be maybe like 10 days. And then I've read stuff where they say 6 weeks, foragers live and work for 6 weeks. Well, it's actually more related to a mileage limit. I think it was ABC XYZ of bee culture, around 480 miles of foraging. And there's a point where I saw another study where the muscles in the thorax lose their ability to take up the glycogen. So there's like it's an aging thing and kind of a mileage limit. So think of it as dependent on the environment. So let's say Manitoba, very long days, middle of summer, canola flow. By 5 a.m. there's a full on honey flow till like 10 in the evening. Those bees probably do only last 10 days. I'm throwing a number out, I don't know, but compare that to like say our coastal areas here with this marine environment where we have 3 or 4 days or a week of rain. The bee, they're not foraging, they're just hanging out. So anyways, I'm getting carried away, but 3 weeks, let's go with 3 weeks, and you can adjust that if you have a different idea about it. So what I'm getting at is these bees that resulted from back here don't get into the honey flow. So now we can walk or animate this along. And so first part of May, those eggs that were laid there, they're in all of the honey flow if there's 3 weeks there. Same with middle of May. And now let's come forward to June, first week of June, 2nd of June. I'm going to cage the queen. Am I really going to miss the bees that would result from eggs laid on June 2nd? What are these bees doing? These are the dearth foragers and the robbers, right? We have into July and August where the robbing can be so bad. Do we actually need that many bees? So you can see how this can be, if it's timed right, it can actually provide like a peak population for the honey flow, and then maybe help you out on the other end where you don't really need all those bees. So winter bee development in August and September. So again, we've got a timeline across the top: June, July, August, September, October. And these are weekly dates. And so our caging was back here in June. And the queen was released around the 3rd week of June. So picture like back in May, we had this peak size of a brood nest, and all of those bees emerged, and now that nest is all cleared out, and we're going to release the queen, and she's going to build that back up and get ready for winter. So they actually only need like 6 weeks or 8 weeks to build that nest up. So you can see there's plenty of time. We've got like say a week in June, the month of July, part of August, and that nest is back up to size. And it kind of seems like the queens sort of make up for lost time in a way, and they really do build that up. So now the important point here, so we have this period that is a critical developmental period. And what we've done is control the mites back in June. And this is so important. You want to be done with mite control by the end of June. You don't want to be like checking for mites and doing treatments in July and August or checking in September. You are very likely to have a problem if you're not done with your treatment and have it all under control by the end of June. So here's an overlay. This is from Randy Oliver's Excel program, where you can do a mite wash and get your number mites in a sample of 300 mites. And so this was like a nuke, a spring nuke. And so look, these numbers along the top, the 5, 7, 10, 14, 20, and so on, that's the number of mites in your wash. So you do like an alcohol wash or a soap wash, and 5 mites. Well, that doesn't sound out of control. 5 mites, 7 mites. But look at these numbers down below. These are like estimates of actually how many mites are in the brood or in the colony. So 1,500 mites, 2,000, 3,000, 5,000, 6,000, 8,000, 10,000, and this colony is going to collapse around sometime in October. And you see this acceleration of this mite growth right in this critical development period. So the message is be done with your mite control in the end of June. Okay, and this is where this brood break is so perfect, because if especially, I know there's parts of the countries where different honey flows and stuff, it's may not work, or there may be issues, but I think from what I know so far, for us in the Pacific Northwest, where we gear everything for this blackberry flow, I think it's kind of the perfect, or can be part of a perfect program. So anyway, so here, now let's kind of look at this development of the winter bees. So I'm going to kind of animate forward here. So if you look at this triangle here, winter bees emerging from the brood. So the way that the winter bees emerge, it's not like all at once in September or October or all of the last bees that emerge, but it's a progressive starting probably for us in August, and I'll show you some data on how I, what I base that on. So let's go up here to July 28th, and there's the queen has laid up, let's just picture a frame of brood, okay, on the 28th. And those bees will emerge from those eggs around the 18th. So a small percentage of the bees that are emerging there will be winter bees. And then as we walk forward or move forward into August, more and more, or a larger percentage of the bees that emerge will be winter bees, and it's kind of progressive. So, and then that last batch that emerges when they shut down brood, that has the highest percentage of winter bees. So you can see, like when I back this up, the bees that start emerging in the latter part of July are the nurse bees that are going to raise these winter bees. So I think you probably get the picture. This is really important. And if you can control the mites back here, you've eliminated that mite load and the viral load, and you have your best chance of having healthy winter bees. So this is one colony last fall, hive number 2. This is the broodminder reading. So this is the temperature reading in the brood. So 60, 70, 80, 90, and 100. So all spring and summer and into early fall, that brood temperature runs right like pretty much flat line on around 94 degrees. And in this colony, the queen stopped laying around the end of the 3rd week of August. So down along the bottom is the timeline. So June, July, August, September, October, November, December. So she quit laying. All the brood emerged mid-September. And I did an inspection on September 30th. There is no brood in the nest. No cap brood, no eggs, no larvae. And so my winter population had emerged in mid-September. Now, this colony shut down earlier than the others. The others like maybe a few weeks later. So every colony is going to be different. But the point is that brood winter bee population could be emerging in August and September. So now kind of changing the subject and jumping to kind of a description of the classical trapping comb. So this is like what they've been doing in Germany back in the 80s. There are beekeepers that since this process was first developed, this is all they do. They don't do chemical treatments. Now, you have to be highly motivated to treat, chemical free to do this because it is extra work. But so I'm just going to kind of quickly go through and let's see, I'm going to take a look at the time real quick here. Okay. Yeah, so I'm just going to kind of quickly go through this. It's recorded, so if I go, too fast and you want to come back and look, you can, pause it and look at these pages. But so day one, you find the queen and you put her on the trapping comb number 1. And I'll point out that that this is one of the issues. Your like peak population, and you want to be comfortable with going in and finding the queen in that time, early June. There, it's not that easy, but I kind of enjoy it. But it is part of the process. You want to be able to handle the queens and some experience handling queens. So if you're new to beekeeping and you haven't handled queens a lot, I would be like, look more at the Italian 25-day caging, because there's less of this labor and less handling. But with that said, day 9, you're going to transfer the queen to the trapping comb number 2. Trapping comb number 1, you're going to put it back in the hive because it needs to attract the mites and be capped. So it's setting outside of the frame cage. You need to look for queen cells, because what I'm finding is that I'm doing this right at when these colonies, I'm trying to grow them and peak them in population for the blackberry, and there's a lot of bees in there, and they're like ready to swarm. So on a day 9, I go through every frame and find queen cells and take them out. And I did a colony today, and they had made some queen cells on the trapping comb too. So now once I go through and do all of that, then it's easier after this day 9, because I don't have to worry about swarming, right? Day 18, transfer the queen to trapping comb number 3. Place trapping comb number 2 next to the frame cage so it collects the mites and gets capped. Remove trapping comb number 1 and I freeze it. Basically, you're gonna you kill the mites and you kill the infested brood. So that was day 18. And this is what it looks like on day 18. Here's trapping comb number 1. This is going to go in the freezer. The queen is in here on trapping comb number 2. This is the frame cage. Trapping comb number 2 is going to go back in here because all that brood is open. It needs to stay there to attract the mites and get capped. Then I'm going to put the queen on trapping comb number 3 in the frame cage, and it goes back in there. So that's like a picture of day 18, if that might help. So day 27, you remove the cage, you're done with it, release the queen, leave trapping comb number 3 in the brood nest to trap mites, remove trapping comb number 2 and put it in the freezer. And then you do have the option of oxalic acid sublimation. So that, again, this would be a modification of the classical trapping comb. If you decide, hey, I think I need to because of the mite load or something, you might and decide to do that. And this research, one of the research projects that I did that we're going to look at was this modification where I did the trapping combs, and I actually did 4 trapping combs. So the queen was released on day 36, and I combined this process with the extended release oxalic acid based on Randy Oliver's work. So day 36, remove trapping comb number 3 and freeze it. Okay, so you can do this with 7-day sequence. Say you only work your bees on weekends. So you add another trapping comb. And so you're in day 1, 7, 14, 21, 28, and 36. So you can adjust this and make it work for the weekends. So staying with this same subject of the classical trapping comb, this is just another way to visualize what's going on. And here across the top is the timeline. So day 1, day 9, day 18, day 27, and 36, just like we just looked at. And this animation kind of shows you when you get kind of a visual here. So now you're on this last trapping comb. All of the mites have emerged. They're all in the dispersal stage. They have nowhere to go. They even though there's some eggs and open larvae over here, it's not mature enough. So they're all attracted to this by scent to this trapping comb. And this is what has happened all the way along, but this kind of gives you a graphic of what you end up with. There's just no place for the mites to go. So this can be like 95% effective at taking the mites out. And Dr. Buchler and his home apiary, most years he never has to use oxalic acid or any Varroacide. He monitors really close, and once in a while he'll get a year where he'll do oxalic acid. So that's just day 36. So so I bought a nuke last year, May of 2020, and I wanted to try a Caucasian queen. So this was an open mated daughter from one of Sue's inseminated queens. And this nuke came from out of state. A couple days after I installed the nuke, I did a sticky board count and there were like 14 mites. This is like a 5 medium frames. So the next day I did a sticky board count and it was 19 mites. Well, that's a lot of mites. And I knew right away, like, I had a problem. And so I've been studying all of Randy's work. If we get time, I'll show you Randy's website and how to get to his papers, but he's been writing on this for 5 years. I'll just quickly, while I'm thinking of it, make a couple comments about what I've learned about the extended release. I haven't played with or I should say experimented with the sponges. I'm kind of waiting to see how that pans out. I had really good luck with the shop towels. And my idea was, what I noticed is the bees work the edges. So the more edges, the better. And so I make these slits in here. And so I just decided to stick with what was working. The other thing that I do, after a couple of weeks, it looks kind of dry, I just pull it out and throw a new one in. And it needs to be moist. And you're not like So let's say if I put 18 grams in and 2 weeks later I pull it out, those towels, probably almost every one of those 18 grams is still there. They make very little contact with it and take very little acid out. And I've taken like a little tiny patch of it that they threw out the entrance, so all dry, put a little distilled water and check the acidity, and it's still like less than 1 on a pH scale. So anyways, those are the things that I learned about the extended release with the shop towels. Just replace it if it looks dry. And you can see they don't, up on these surfaces, the top surface, they don't hardly contact that at all. They work along the edges and at their own pace, and they're like dosing themselves basically. So this is all the data that I put together from that colony that I was talking about, the nuke. So let's, this again, classical trapping comb combined with the extended release oxalic acid. And so this is a timeline along the bottom horizontally. We got May, June, July, August, September. These are the days of the, or the numbers of the weeks. Each week had a number. Week number 1, number 2, number 3, number 4. And these are weekly mite counts. So the mite kill or the drop is along this vertical axis. So you can see 100, 150, 200, 250, 300. So what else? Okay, so these blue symbols with the numbers, that's the extended release with the dose. So week 1, there was a dose of 9 grams, another week 5, 9 grams, week 8, 36 grams. These orange colors are when the trapping comb was pulled out of the colony. So there was 4 of those. Mite washes, there's a mite wash here, 9 mites out of 400 bees, so 2% infestation. Then there's another mite wash, 15 mites in a sample of 441 bees. So we're up above 3%, 3.4% infestation. And then there's a mite wash here, October 1st, which had 1 mite in it. So I should say, like when I started back in May and I saw those mites in there, I didn't like plan this all out. Like, okay, I know exactly what I'm going to do. It actually, I don't claim to know exactly what I'm doing, but what I decided I'm going to, I put 9 grams in right away. And you can see it definitely killed some mites. And I did this mite, I should have done a mite wash on day 1 and I didn't. So if I would have, would have a better idea of the actual number of mites, maybe, but we'll get to that later. The starting mite population in there. But so got into June and I'm starting to wonder, like, am I really getting ahead of this? I put another 9 grams in, and then a few weeks later I did a mite wash, and you can see that the mite count went up. So at this point I'm thinking, you know, I don't really know if I'm getting ahead of these mites, and I'm not going to mess around. So I put 36 grams in, and I started 1st of July, the trapping comb, and I did a sequence of 4 trapping combs. So you can see what happened here. Right away, this mite drop goes way up. And and of course, 36 grams of oxalic acid extended release. It's the combination of the brood break with the oxalic acid extended release. Because if I wouldn't have done the brood break, I would not have been able to get this kind of kill. So basically what I did is by caging the queen, I forced all of the mites out of the brood. And that quickly, within a few weeks, you can see what happened. I'm in August down to like counting, most of my daily counts are like 0. So this is like a triple whammy. Oxalic acid extended release. The brood break, and any mites that are getting past the oxalic acid and are getting into the brood are getting hauled out and put in the freezer. So I'm just gonna go through this really quick because this is just the data that I use to build that chart. So this is May 20th. This is the calendar for the month. Nuke was installed on the 9th. This is the oxalic acid on the 15th. These green symbols are the weekly mite counts. So 155 that week, 314 that week, 171. And then in the black square in the corner here is the running total. So you can see, like, here I am barely 2 weeks into this, and I've killed almost getting, over 450. By this 3rd week, I'm over 600 mites. So I'm guessing there were probably like maybe 500 mites in there. So anyways, yeah, so these red numbers, those are the daily mite counts. And then that's June. Here's July. So here's where I started the first trapping comb queen cage on July 1st, 36 grams on the 3rd. And then, it's just the rest of the data, and I got to move this along. So you can see in September, even back here in August, most of these counts now are 0, and then there's a 1 and a 3 here and there. So basically pulled the mites out of there. And here's from Randy Oliver's website, scientificbeekeeping.com. He's got this mite model. Hopefully everybody is familiar with this. He has some videos that will show how you use this. This is amazing, amazing tool. I just love what he's done here. But so what this is a spring nuke starting with a population of 500 mites. It's crashing in September with 17,976 mites, give or take a few mites. So if I had more time, I'd tell you more about this mite model. This is an amazing tool. So now we're going to jump forward to December. Same colony, and 2 oxalic acid sublimations 12 days apart on December broodless colonies. So December 2nd, so this is the mite kill here, and this is the timeline along the days of the month. December 1st, December 2nd. 2 grams oxalic acid sublimation. And then coming back on 12 days later on the 14th, 2 grams again. So basically this shows you that there's basically hardly any mites left. There were 7 mites in this first kill, and then I got 1 mite on this second kill. So the point is, not a lot of mites were left after I did that trapping comb with the extended release and the brood break. So then I want to make a point about this. So when you do your December sublimation with oxalic, the standard is 1 time, usually in December. It could be November, once you know they're broodless. But I think this standard might be changing. Last fall, I heard Bob Binney say that he's going through and doing 2 treatments when they're broodless. For him, I think it was in November. And he was saying that evidently other commercial beekeepers are doing this because it really is a lot more effective than the single dose. And I think there's some research going on now. And this was my little attempt at researching that idea. So this is my whole apiary. So 8 colonies in December, they're broodless. So the total number of mites that I killed, 959 mites. That first treatment got 659 mites, and the second treatment, there were still 300 mites that I took out with that second treatment. So I just want to throw that out. Keep an eye out for research, and maybe think about if you need to, or if you just want to be safe, do that extra treatment. So yeah, so queen number 3, this is that queen. And a few things to kind of think about. So, you know, some people, the first thing they'll say is, well, you got a colony that was full of mites, so you know, you should get rid of that queen. But, you know, we don't know the history on the makeup of that nuke, for one thing. They're probably throwing, you know, a frame from here, a frame for there, and then dropping a queen in. It may probably had nothing to do with this queen. And in fact, I'm convinced, because this queen, she wintered, came out of winter, took off like crazy, has a huge colony. She's on a trapping comb now. I'm doing the same thing, a series of 3 trapping combs. And so you saw like in December, almost no mites. And then the few that were left, I took those out. So random mite drop counts, January, February, March, April, May, 0 mites. I never picked up 1 mite. And then I think June 2nd, I did a mite wash, 0 mites. So, you know, if you give them a chance and control the mites and don't let the mites get ahead, I think you don't blame it on the queen, unless you're really convinced. So that's one thing to think about. The other thing is that you saw like what was it, like 3,000 and something mites, and you're thinking, oh man, like that's a serious infestation. But if you look at those mite counts, they're like the wash, one wash in I think that was July, 2%, and then it went up to 3%. That this queen stayed ahead. She laid like crazy. She built out from 5 frame nukes into like 3 mediums, and then I think I had a super on when I started the caging. So she laid like crazy and stayed ahead. I did early in May and first part of June. I did see a few bees. I had a little platform out front so I could kind of see what they're throwing out, the entrance. And I did see a little bit of deformed wing virus, but not very much at all. And they got it, basically, like I say, they got ahead of it. So I'm gonna quickly go through a few other slides because there's a couple things I want to show. So I did have a permit from the state. I want to kind of point that out because the oxalic acid extended release is not approved yet. I had to write a label, but basically it was just all based on Randy Oliver's research. So I'm really not doing anything original here, except I haven't seen any research where the brood break and the extended release oxalic acid has been combined. So that is, maybe that's something new. I'd like to see more research, and if anyone knows of research on that, I'd like to know about it, because I think it's like a very powerful combination. With extended release, Randy's idea, and he's commercial, right? He wants to go in and throw the sponge in there and walk away and leave it in for, you kind of have to leave it in like 2 or 3 brood cycles to get the effect, but you still end up with these outliers. And there's I think by combining it with the brood break, you're going to improve the efficacy, and you're going to shorten that range where it's like a tighter efficacy, and not so many of these outliers. I think it's going to be pretty powerful. So Randy Oliver, scientificbeekeeping.com. Hopefully everybody knows about that. What I want to point out is go over here on the right where it says articles by publication date. Everything that he's written over the years, it's in order, like the most recent to the oldest. So you can go back through the years and find like the articles that he's written on the extended release. You go back 5 years. And if you're interested in extended release, I really recommend that you study all of this and kind of be, educate yourself and be up to date on it. And also, so there's a link on that page on how to get to the Mite model. And so check that out if you don't know about it. Okay, so real quickly on equipment. So there's, and this is where, like in the beginning, I said, queen caging, 25 days, like they do in Italy, release the queen, oxalic acid. There's the German classical trapping home, but there's so many ways to do this, different types of cages, and so it's something you just have to look at. this is on Bob Binney's YouTube channel. He came out recently with this. This is how to build this queen grafting timing box. This is perfect if you can do something like this. So check that video out. In Europe, on the market, you can buy these. I found this is in Spain. I have no idea what it would cost to have one shipped. But that looks like a pretty cool setup there for a cage. I had to make my own with sheet metal, and since I'm all on mediums, even if there's just no equipment to buy. So that's another drawback to this, is like the equipment isn't available. There are no mentors that are doing it, so nobody knows how to do it. So there are things that make it more difficult. That's the trapping comb. Now, the 25-day queen caging, Italy. Okay, so I showed you the cage, the little tiny cage, but you can cage. I did 2 years ago, I just made a cage. I ripped some frame parts and made a real narrow cage, and queen caged in that for 25 days. So you can, so my buddy Greg shared these pictures with me. Like I said, he's been doing this for a couple of years, and so this is just, you cut out in the frame, and this mounts inside, and it's got a little door there. Pretty slick setup. Like I say, in Italy, they do this. They just keep these in the hives year round and use them a couple times a year. And there's a picture that Greg shared with me there. Okay, I think that's the end. I'm going to stop sharing, I guess.
Thank you very much, Rick.
What can one say?
Okay, so
Yeah, hooray.
I'm in awe.
I hope I didn't bury you, or I started talking about this stuff, and it's a lot of information. But even if you're just a beginner, and you're saying, hey, I'm not going to do this, maybe like in a couple of years I'll think about it. But I think along the way in there's things that are educational, so hopefully it helped everybody. So I guess let's do some questions. Is that what we want to do? Eli?
Yeah, actually, there's a couple things. It sounds like the 25-day one requires less queen handling. So
Yeah.
if you're not comfortable handling queens, it sounds like that's one people would go with. So I kind of wanted people to sort of think about that. The brood minder, you had a picture of one of the strips, and
Yeah.
those are temperature sensor strips that are placed inside the hive. And that was the green chart that Rex showed. So you can look that up, and it's kind of fun because you upload the readings on your cell phone. You just stand next to those strips, and once you're set up, the app uploads it for you, and you can see that change from 68, 70 degrees the queen has to be kept alive in the center of the cluster, to the 92 to 95 degrees for that the brood needs when they're actually raising brood. So you can actually, one of the big questions in the Pacific Northwest is, does the queen ever stop laying, and do they ever stop raising brood when we have a really mild winter? And that answers the question. So that was kind of one of those things. A lot of things, like you said, you touched on, a lot of things a beginner can kind of take out of this. One thing in beginner, I bring up the queen life cycle and these numbers of days. And that at 3 days the egg hatches, the workers, if they decided to make a queen, They are making a queen. She emerges in 13, and then 5 to 6 days till she works with her muscles, eats like a teenager apparently, and gets ready to go on mating flights. And there's all these things that sync with the natural brood break and that what you're trying to imitate. So I hope that the beginners didn't feel totally lost, and every once in a while said, yeah, I remember something about that. And I could, like you said, and I could do this in a couple of years. And maybe my hope is that somebody is going to start a massive project, maybe in Western Washington. Maybe Rex is going to do his PhD and get enough of these cages that people could actually try it. And that increased honey production is the one that would sucker a lot of people in, because if they're not raising brood, did everyone listen carefully to that? If you can do that brood break at the right time in June, everybody's out foraging, nobody's staying home babysitting. And the honey, and this is actually a technique, I don't know what it's called, but it has been used for a lot of years, deliberately removing the queen, caging her. You can take her out for a week or two, but this system with that huge queen cage, I love it. Her pheromones are going to be all over the hive. It is going to work so much better than sticking her in a little dinky thing where she can't spread, because she spreads with her tarsi, and the workers are spreading it all around, and she's going to be in contact with so many more workers. I love that part about this. Okay, I raved enough. Anybody else really have questions? I'm hoping that even beginners saw glimpses of something you could do that will start to make sense, or already does make sense. I think we could do this. I think this is one of the plans that could work. Anyone else?
I have a question.
Hi, Sarah.
Hi. So I think I followed pretty much everything, I think. But one thing I was thinking about was, so when the queen is not permitted to lay wherever she likes, and eventually cells become available, I mean, you must have a different way of talking to your bees, because mine like to backfill like immediately. And so I'm just kind of wondering, like, right as the nectar flow comes on and a lot of these cells are becoming available, do you see them just filling up your entire, you have medium, so would fill up pretty fast, I imagine? Or do you not mind that? It's not a problem.
Yeah, that doesn't seem to be a problem. That you make sure that you have enough. So I'm in mediums, so I have 3 mediums for my brood nest, and then, but I want to make sure I have a couple of supers up above, so they're storing up above, and I really haven't had a issue with that yet. I think the bee, as long as they have room to store that nectar up above, they're going to clear out the nest for the queen to lay. And so I remember last year being thinking about that and wondering if it was going to be a problem, but it didn't seem to be a problem. Did that answer your question?
That's a good question. Yeah.
Yeah. So, yeah, I think as long as they have room, they do tend to want to move stuff out of the way of the queen. So as long as they have room to put it, like if they had empty, open drawn comb in a super above, and then, you don't need to have your excluder in the way to either, because you're already excluding the queen.
Mhm.
So I didn't have a problem with that, but that's a good question. I'll make a quick comment on what Eli, your reaction to the little tiny cage. It's kind of a common reaction, because it just seems like